So it's time for our final NGL talk of the Showcase Symposium. It's my great pleasure to introduce Professor Cate Peña from Princeton University. Cate investigates how environmental experience in early sensitive periods impacts behavior via enduring transcriptional and epigenetic mechanisms. As a graduate student at Columbia University with Francis Champagne, Cate studied how maternal care influenced brain development and behavior. And as a postdoctoral fellow with Eric Nestler at Mount Sinai, she established a two-hit stress paradigm in mice that was ideal for investigating the molecular and cellular mechanisms that mediate sensitization to future stress prior to the onset of its behavioral symptoms. So Cate's ongoing research applies genome-wide sequencing and bioinformatic analysis approaches to investigate the enduring impact of early life stress. So today she'll give a talk titled Lifelong Transcriptional and Epigenetic Mechanisms of Early Life Stress in Brain Reward Circuitry. So please welcome me in welcoming Cate.
Thank you. It's really an honor to be here today, both to share my work with you, to be part of the next, cohort of NGL council members, and to learn from all of you, all of the team talks that have been presented and conversations of posters and in through the evening at dinner have just been really wonderful. And I feel like the future of science is truly happening here, and it's really exciting to get to be a part of that here. So my work is really motivated to understand how early experiences during development are maintained into adulthood to impact behavior. It sounds a little bit like what Keith was interested in, but I'm approaching it from a completely orthogonal point of view. And in particular, I'm interested in understanding how one very salient experience early in development, early life stress, changes brain development to increase risk for depression and other psychiatric disorders. So when we think about depression and other psychiatric syndromes presenting in adulthood, and even just as basic neuroscientists in general, we're really biased to investigating what's happening in the adult brain. And sometimes ignore the events and environment really leading up to that point of development. But I want to challenge everyone here to start thinking about development and what's happening in the adult brain is really a consequence of a lifetime of experiences. And in particular, my work is focused on understanding the mechanisms through which early adversity really increases risk for depression and depression-like behavior across the lifespan. So when we, unfortunately in the United States, when we think about child maltreatment, this affects many individuals. In the US alone, more than 1 million children are impacted by early adversity, including neglect, physical, sexual, or psychological abuse, and other forms of early life stress, including loss of a parent due to death or incarceration, poor parent-child bonding. There's a growing body of literature investigating the impact of experience of natural disasters at different points across the lifespan. But recently, it's become hard to ignore that childhood adversity doesn't even begin to describe it for millions of children worldwide. Children are disproportionately affected by violence and displacement, and the IRC and UNICEF estimate that around 30 million children worldwide are impacted by violence and have been forced to emigrate. And these migrant children are at an increased risk for harm, including malnourishment, deprivation of many kinds, trafficking, and other extreme forms of stress. So it really motivates us to understand how these early adversities in any of these forms, more or less extreme, are impacting brain development to increase these risks. So what are the risks? Well, stress impacts the whole body. Physically, it increases risk for heart disease and hypertension, but I'm really focused on the robust clinical finding that early adversity increases risk for major depression and other mood and drug disorders by more than 2 to 4-fold. Not every individual who experiences early life stress will, of course, go on to develop depression-like behavior or any of these other adversities, but it is associated with heightened sensitivity to stressful events later in life. And luckily, interventions show us these outcomes are environmentally driven. So we can move from a stressful environment into a less stressful, more enriching and supportive environment and reverse some of these negative outcomes. But we think that the early environment is really a sensitive window for some of these experiences to shape brain development and transcriptional patterning, which is what I'm looking at, in the brain to increase these risks. So how do we gain access to brain tissue and model this in a way that we can understand how this is impacting development and sensitivity? And I've chosen to do that in a mouse model in the lab. So the type of early life stress that we're imposing on these animals is a combination of maternal separation stress. For about 4 hours a day, the pups are removed from the home cage into their own cage here. And that's in combination with a reduction in the nesting material, which has been shown by a number of labs to increase disordered maternal care. And in order to test whether there is a sensitive period for early life stress to increase sensitivity to stress later in life, I'm looking at, I've imposed a stress at 2 different time points in postnatal development. These are each 10-day periods, either from P2 to 12, which I'm calling ELS1, or from P10 to 20, which I'm calling ELS2, and I've been able to refine that period a little bit down to P10 to 17 for those who are interested. And of course, to understand whether these experiences early in life increase sensitivity to a second hit of stress later in life, I've subjected half of these animals to social defeat stress for male mice, or a different stress for female mice that I'll get into in just a second here. So the social defeat stress is a very widely used paradigm to induce depression-like behavior, in which an experimental mouse, we use mostly C57s, is forced to intrude on a cage territorialized by a larger, more aggressive CD1 mouse. They have 5 minutes of physical interaction during which the larger, more aggressive mouse physically bullies the experimental mouse. And then the experimental mouse is separated across a semi-permeable plexiglass barrier for the remainder of the day. So they have sensory contact, but no longer physical contact. And this is repeated every day for 10 days with a new bully mouse. And this leads to many aspects of depression-like behavior that we can model from the human disorder as well. In female mice, we use slightly different adult stress. This is a subchronic variable stress or subthreshold variable stress. They're subject to different stresses each day. And I've specifically chosen for the females, which I can talk more about offline later, this different stress at a subthreshold level to try to tease apart this increased vulnerability. So when we're trying to model early life adversity and depression-like behavior in mice, it's important to remember that we're not modeling all aspects of early adversity as we see in humans, and we're not modeling all aspects of depression-like behavior as we see in humans, but we can model endophenotypes. And in particular, the social defeat stress is one of the most used models of depression-like behavior because it recapitulates many aspects of human depression, including social avoidance, anhedonia, anxiety, weight and circadian changes, and is responsive to serotonin-altering drugs in approximately the same 3-week time scale as we see among humans. The primary behavioral measure that we're looking at is the social interaction test, which is able to recapitulate some aspects of social avoidance that we also see in humans with depression. So in this test, an experimental mouse is allowed to explore an empty arena. There's an empty cage at the top of this. Sort of serves as a novel object, and they explore for a few minutes. We remove the experimental mouse and introduce a novel social target that it's never met before in this interaction zone, and the mouse is allowed to explore again. And most mice are very social. They spend a lot of time exploring the novel social target if they have not gone through social defeat stress. However, mice that have experienced social defeat tend to be much more socially avoidant. They do explore the social target to some extent, but they're much more socially avoidant in these corner regions here. As in humans, not all mice can be considered depressed. Some of them are resilient. So if we have control mice, we can see the ratio of time spent exploring the social target to the empty object tends to be greater than 1. There are many mice who show this susceptible-like phenotype, spending more time in the corners, and there are also mice that are resilient. This is important because it allows us to then alter aspects of the environment or the brain to try to shift the proportion of animals considered susceptible or resilient. And that's exactly what we're looking at here in this early life stress paradigm as well. So if we're just focusing for now on animals that have experienced either standard rearing conditions or early life stress in either of these 2 postnatal periods without experience of adult stress, just the control conditions, what was somewhat surprising at first is that all these animals are still very social. And you can see this in the heat maps of their behavior as well. They spend a lot of time exploring the novel social targets, even after experience of early life stress. So early life stress alone in either time period does not really seem to alter depression-like behavior. And this is just one of a number of tests of depression-like behavior that I'm showing you right now. However, after a second hit of stress in adulthood, animals that experienced early life stress, particularly in this later postnatal period, this ELS2 period, have dramatically reduced social interaction or increased social avoidance, which again you can see in these heat maps of their behavior. So there's some kind of latent sensitivity to this stress later in life, or an increased sensitivity conferred by the early life stress in that later postnatal period. So I won't go into the whole battery of different tests of depression-like behavior that I've done for both the male and female mice, but I've sort of generated a composite depression-like behavioral score. Once again, showing you that for both male and female mice, those who've experienced only control conditions in adulthood don't have a very high proportion of depression-like behaviors. But after that second hit of stress in adulthood, particularly animals that experienced early life stress in that later postnatal period, both for males and for females, have increased depression-like behavior. And this really indicates that there is a novel sensitive window that we can kind of tease apart for early experiences to increase sensitivity to depression-like behavior. And it also allows us to model and understand the mechanisms through which early adversity increases this vulnerability or sensitivity later in life, even prior to the onset of these behavioral differences, which is pretty cool. So this generates a whole bunch of questions that we can now ask in the brain, and I'm just going to take you through two of the questions that I had worked on during my postdoc, and then just a few slides at the very end of some of the ongoing work that I'm exploring. So one of the first questions that we addressed was, how does early life stress alter long-lasting transcriptional patterns in key brain reward areas? And is there a sensitive period, like we see for the behavior, for this predicted transcriptional regulator that came out of a lot of these transcriptional analyses, OTX2, which I'll talk more about, to mediate this behavioral and transcriptional stress sensitivity? And then finally, the ongoing work I'll share is exploring the chromatin mechanisms that might maintain this long-lasting transcriptional priming. Okay, so first, how does early life stress change long-lasting transcriptional patterns in key brain reward circuitry? And I'm really focused on the brain reward circuitry, including ventral tegmental area, which is the dopaminergic hub of the brain, projecting to nucleus accumbens and prefrontal cortex, because this circuitry underlies many aspects of depression-like behavior that we can model in the animals, including anhedonia and social avoidance. And it's also key to some of the other increased risk syndromes, such as drug abuse, that we see in humans and also in the animal paradigms after early life adversity. We first wanted to look at the transcriptional changes happening, these long-lasting transcriptional changes in the adult brain. So I collected 163 samples in a 2x2 design of early life and adult stress across these three regions, and in males also a ventral hippocampal region, from both male and female mice. So normally when I present this, people are impressed. That seems like a lot of samples among this audience. It really pales in comparison to the scale at which you're performing science, but hopefully you guys are still impressed from a single individual's work. And we started doing the sequencing in 2013. It was bulk tissue sequencing, poly-A tail. And I've been wading through all of the analysis over the years, and I'm excited to share some of the conclusions that we're able to draw here today. And in particular, most of the analysis I'll show you today is really focused on the ventral tegmental area, although we're going back and looking at all these different brain regions, again, in males and females. Okay, so first, just to get sort of a high-level overview of what was happening after early life stress, adult stress, or the combination of the two stresses, all compared to the same standard rate control group. This is a union heat map, yellow indicating increased gene expression, blue indicating decreased gene expression. So just a very basic overview. We're setting a 20% fold change and an uncorrected p.05 cutoff criteria here. And what was immediately apparent is that early life stress really seemed to induce somewhat similar transcriptional changes as adult stress or the combination of stresses. This was even more striking to me because these brains were harvested in adulthood. So this transcriptional signature that seems to be similar to what's happening in the adult brain is a very long-lasting signature. And even beyond that, these animals are not yet showing changes in their behavior, yet they're showing a similar transcriptional pattern. So to me, this sort of provided evidence for a depression-like transcriptional state that emerges even prior to these changes in depression-like behavior. So this is looking at it in sort of a thresholded way. Again, most of the analysis I'll focus on is looking at the ventral tegmental area, and I wanted to also take a look and see what patterns might emerge in a non-thresholded way. So I used rank-rank hypergeometric overlap analysis, which is a threshold-free way of comparing or evaluating similar effects across conditions. So we can have one comparison here, for example, early life stress transcriptional patterns compared to standard control, and another comparison on the other axis, for example, adult stress versus standard control. And we're using a quadranted version of this analysis, so we can see similarities between these comparisons in genes that are upregulated by both, downregulated by both, or oppositely regulated across comparisons. And again, in the VTA, in male and female animals. And first, what we see when we compare early life stress to what's happening after adult stress, as I showed you in the heat maps already, these conditions induce very similar transcriptional changes. This is very interesting and supports, again, this idea that maybe early life stress is already inducing this depression-like transcriptional signature. However, I next wanted to know what was happening after a second hit of stress. Or in other words, what does stress in adulthood do depending on your history of early life stress, whether you had early life stress previously or not. And here we see a very different pattern. There's absolutely no similarity. And I'm still working my way through trying to understand how this pattern is being maintained and regulated and how that maps onto behavior. But for now, I've really focused, we started with this. What is this transcriptional priming potentially doing, and how is it being regulated in the brain to change behavior? So I use ingenuity pathway analysis, which is a tool put out by Cate, and it's a data-driven way from known biological interactions, predicting what upstream regulators might be responsible for some of the changes in gene expression and these patterns that we see. And top among these, at least in the build in 2014, was OTX2. And I became really interested in this for a number of different reasons. So here's OTX2 and the genes that had led to its initial prediction. What is it? It's orthodentical homeobox protein 2. It's a transcription factor that binds to a canonical bicoid sequence. It's highly involved in brain patterning and midbrain patterning, and in particular, dopamine neuron development. So again, we're excited by this. We're definitely in the right region here in the ventral tegmental area. And it gained notoriety about a decade ago now for its role in critical period plasticity in the visual system. So I know many of you here are working on visual system and plasticity. You might recognize OTX2 from Takao Hensch's work in this area. And it was also very interesting because in the visual system, it's not transcribed. The mRNA is not transcribed in V1, and it seems to be the protein seems to be transported across synapses from potentially retina and LGN all the way to V1. And then finally, OTX2 is potentially involved in epigenetic enhancer regulation during neuronal differentiation, which I'll come back to sort of at the very end. This is a very different time point than what I'm looking at in postnatal development, but still seems very interesting in its potential role in a lot of different aspects of brain development. So one of the first things we wanted to do, especially after the implications for a parakine mechanism in the visual circuitry, was understand whether OTX2 was actually itself transcribed in the ventral tegmental area. And the absolute first thing I did was to turn to the Ellen Developing Mouse Brain Atlas. And in fact, OTX2 is transcribed in the ventral tegmental area. This is just at P28, but we can see it across development. And also interesting is another study that showed that the protein is restricted to the ventral tegmental area and not to substantia nigra in the adult brain, implicating a role in more limbic regulation than motor regulation. Okay, so looking at the target genes that had led to OTX2's prediction, all of these are downregulated by early life stress in the adult brain. Once again, very long-lasting changes. And even more interesting was that after adult stress or after the two hits of stress together, the transcript is also downregulated. Again, sort of indicating that this might be a master regulator of this depression-like transcriptional state. Let's see. Oh, I wanted to go back and point out that you might notice here OTX2 itself is not altered in the adult brain. So this really kind of scratched the back of my brain for a long time. I didn't understand how OTX2 could possibly be having such long-lasting effects on transcription if this really is an interesting driver to go after, if it itself is not altered in the adult brain. But I'm really interested in time courses just in general, and I thought, well, let's go back and see whether OTX2 is altered by early life stress acutely at the time of stress. So just using qPCR, I built a time course. This is in the standard reared control animals. It's very highly expressed early in development and decreases a little bit with development, which you can see reflected here in the standard reared animals. This is before early life stress, immediately after, in adolescence and adulthood. So what happens with early life stress in that later postnatal period? And what we saw, which was very interesting, was that ELS2 transiently suppresses levels of OTX2, and there are no longer group differences by adolescence and adulthood. So then, of course, we wanted to know, well, what happens if we look at the brain after early life stress in that first postnatal period? And in fact, we don't see these same differences. You might expect to see suppression right after the ELS1 period at P10, but we don't even see that. So somehow we see this specific sensitization to later stress behaviorally when stress is experienced in this late postnatal period. We also see that specific transient suppression in OTX2. So we got pretty excited about this. And the next question is, well, can we modify it? Can we mimic or rescue these effects? And I turned to herpes simplex viruses. So most of the time when you're doing a developmental study, you want to use something like AAV, very long-lasting. But here we actually took advantage of the very short time course of the HSV virus. So it comes on very quickly within a few hours of infection, reaches maximal expression about 3 to 4 days, and then turns off by about 8 to 10 days after infection. So we generated 2 vectors to either overexpress OTX2 or knock down OTX2 in the brain. And one of the first things that I'll show here is we wanted to ask whether OTX2 overexpression, just a transient overexpression, could rescue this enhanced vulnerability to depression-like behavior. So animals went through early life stress, and then at P15, I overexpressed OTX2. And then the group was later allowed to mature into adulthood and experience social defeat stress here. So just focusing for now on the green bars where we had the control GFP overexpression, we recapitulate the same effect I showed earlier where this increased depression-like behavior, decreased social interaction. And then there seemed to be this overall effect of OTX2 increasing social interaction or decreasing depression-like behavior. And even more exciting, it's always kind of unexpected when things work out so nicely, we were even able to rescue the expression of some of the genes that had led to OTX2's prediction in the first place. And again, this is all looking in adulthood after a transient overexpression. So somehow these are very long-lasting changes, even though the manipulation is no longer happening in the adult brain. And next, I wanted to know whether we could mimic the effects of early life stress by transiently knocking down OTX2 during development. So here, again, at P15, I knocked down OTX2. Animals were allowed to mature into adulthood. And they went through a subthreshold level of social defeat here, where we don't normally see changes in behavior. And once again, which is very exciting, we were able, even with this just transient knockdown of OTX2, to recapitulate the effects of early life stress and see that decreased social interaction. But this doesn't tell us anything about a sensitive period. So next, I performed this exact same experiment, but all in adulthood. So OTX2 was knocked down in the adult brain. I had a 6-week wait, and then again went through subthreshold levels of social defeat. And we don't see any differences, which really indicates that there's a sensitive window for the long-lasting effects of OTX2 manipulation on brain development. So to just briefly summarize what I've shown you from these first 2 parts before moving into some of the ongoing work, early life stress primes the brain to be in this depression-like transcriptional state, even before the onset of depression-like behavior. Stress in adulthood has a distinct transcriptional signature in the brain, depending on the history of early life stress, and this is really work that I'm trying to understand more deeply moving forward in my lab. OTX2 seems to be an upstream regulator and is transiently suppressed by early life stress. And there is this juvenile sensitive period for the effects of OTX2, which we're able to bidirectionally manipulate. And I wanted to stop just a bit here and really say that this whole pipeline really brings together the power of animal models and big data in a lot of ways. So we're able to model some aspects of early life stress and some aspects of depression-like behavior in animals, harvest brain tissue, and do really complex analyses at different levels. And there are many different types of analyses to even do from here with all the data we've collected. But the point is that we're able to generate biologically meaningful hypotheses and then test these back in the animal model, looking at their behavior. And of course, the hope is that these predictions and modeling will have some impact on our understanding of vulnerability in the humans. And in fact, recently, a collaborator, Joan Kaufman at Johns Hopkins, published a paper taking some of these findings, looking at OTX2 as a potential biomarker in children who had experienced early adversity, and found that it's even related to aspects of brain connectivity in these children who've experienced adversity. So it's really exciting to see this pipeline all the way come from the animal model and then be meaningfully related to what we see in humans. So just in a couple sites here, just to pitch what I'm looking forward to doing most and working on next, I wanted to understand what chromatin mechanisms might maintain these long-lasting transcriptional priming effects. So one of the things that maybe didn't pan out in a nice parsimonious way at least, OTX2 seems to be related to sort of initiating these effects that we see, long-lasting transcriptional effects, but does not seem to be related to maintaining them, and I can go into that more offline later. So I wanted to know what might be maintaining these effects, and I turned to understanding what might be happening at the level of chromatin. So here's just a cartoon of the histone proteins and the tails and all the different modifications that are possible on these histone protein tails. They can be monomethylated, ditrimethylated, acetylated, et cetera. And in collaboration with Ian Mays' lab at Mount Sinai and Ben Garcia's lab at Penn, we've isolated these histone proteins, broken up the histone tails into these tiny little chunks, and performed mass spec on these chunks to get an idea of the proportions at each of these residues of the histone, of the chromatin modifications that are occurring. And this is in adult VTA, so long-lasting changes. And just to summarize all the different changes that we've seen, much to my surprise, the majority of these changes that we see in the adult VTA are associated with a permissive transcriptional state. A very small proportion associated with a repressive state, and then a few associated with a priming state, which I became very interested in. And in particular, this included H3K4 monomethylation and H3K9 trimethylation in conjunction with K14 acetylation. And these are both upregulated after, or enriched after early life stress in the adult VTA. So why might we be excited about gene priming in particular? Genes need to be expressed, and they have an active promoter, they have enhancers, and they exist in a 3D conformational state. And the enhancers can either be active or primed. And H3K4 monomethylation is one of the markers of these primed enhancers. So I've done chip sequencing for H3K4 monomethylation at this point. I'm working on a collaboration with some published high-C chromatin data to get an idea of this actual looping structure and what these regions might be contacting in terms of gene expression. The idea is to put these two analyses together with the RNA sequencing data and look for regions of enriched priming and determine the contacted genes that might be more reactive to future stress. And we already have some promising results from this. So to just summarize this last bit, we do see that early life stress increases chromatin modifications in the VTA associated with open chromatin, potentially making it more reactive to future events at the transcriptional level. So we see at the behavioral level, animals that experience early life stress are more sensitive and reactive to additional stress, and we actually see this reflected at the transcriptional and epigenetic level as well. And this might be mediated by K4 monomethylation. So of course I want to look at this in other ways, looking at different mechanism or measures of chromatin state, including attack sequencing. And then again, the beauty of these animal models is we can test these predictions back in vivo, manipulate K4 monomethylation levels, for example, and test the causal impact on behavior. I didn't do this alone. I did this with an amazing team of people. I want to thank my postdoctoral advisor, Eric Nessler, for really letting me run with these projects. He's an amazing mentor. Many of the people in the lab contributed to different aspects of this work. Collaborators in Li Shen's lab have helped with some of the bioinformatic analysis. Ian Mays and Ben Garcia, as I mentioned, helped with some of the mass spec proteomics. Rachel Nevy packages our HSV viruses, and Sergio is helping me put together the high-C chromatin data. And thank you all for your attention.
Great, we have a few minutes for questions.
Actually, I have a question. I was interested in a couple of things. Really great talk. Did you see any structural changes in either VTA or prefrontal cortex in the animals that were most affected? Or
So we looked at gross numbers of dopamine neurons that don't seem to be changed. One of the things I'm going after now is very, very preliminary data that the projections might be altered from VTA to PFC and from VTA to accumbens. And a collaborator at Penn also sees something similar in human imaging. So we're hoping to replicate that and put it together, which is cool.
I was also interested in the animals that were resilient. Did you see any transcriptional signature of resilience or even epigenetic signature of that?
Yeah, it's a great question. I don't have the power to look at that really well right now, so I've clumped the susceptible and resilient animals together. I do have the individual signatures to go back and look, and just as sort of like a basic understanding, when we do cluster analysis, they don't seem to cluster together. They tend to cluster better with their group than with the resilient or susceptible portion, but again, since it changes the proportion of animals that are considered susceptible, we lose the power to do that very nicely.
Yeah, this is amazing talk, really amazing work. I have many questions for you, but maybe one quickly, yeah. So have there been any studies on OTX, one or two? Which one is it? Two. OTX2 in correlations with depression in humans. For example, polymorphisms that are associated with vulnerability to depression, or actually, yeah.
Yeah, so not polymorphisms, but I mentioned this study by Joan Kaufman's group, and she was inspired by this work actually, and she has a huge cohort of children who were either control or had experienced adversity, and they've done imaging in the brains in adolescents, and then they had buccal swabs, I believe it was, where they looked at DNA methylation and a 450 KRA, and she found that OTX2 was related to adversity and to changes in the connectivity they see in the children. So to me, that's very exciting that this might pan out in a human population at all, even in a very different sort of manner. But
what was the biomarker? How was it assayed? What did you say? It was DNA methylation, but from actually the mouth.
Very different, yes. So we can discuss that more offline.
Yeah. And I hope you will be moving to single cell realm or single cell world.
I would like to as well. Yeah,
Okay.
There's a couple projects in the pipelines where I think that single cell is the best way to go, but I'm speaking with as many of you as possible to figure out exactly the best way to approach that.
I'd be happy to help.
Thank you.
Hi. More of a general question about using animal models for looking at long-term protein expression. How well does comparing an endurance in protein expression over 6 weeks really equate up to looking at like a child at 5 and the protein expression at 18? Because even the turnover rates, I would think, over such a short period could have a confounding effect.
So I don't necessarily think of it as stable protein necessarily, as these long-lasting changes in transcription, which can be mediated by stable chromatin modifications, for example, and you just continually have the same level of protein output. But it would be an interesting question to see what the fluctuations are over development and to try to label single proteins potentially even.
Thank you for a fantastic talk. I'm wondering, because back in the day, people have also done studies on stress levels in pregnant mothers and how that can have an impact on the ability to cope with stress down the road. Do you think, or do you have any insights into whether the mechanisms might be similar or could they be different? Because there, obviously, the implication was that the cortisol affects brain development. So are you also looking sort of at what might be regulating the function of OTX2 in that process?
So how OTX2 relates to prenatal stress as well. There's a huge, great prenatal stress literature. It's getting bigger and bigger. I'll point you to a lot of Tracy Bale's work. She's looking at how the placenta might help regulate some of these effects. As far as I know, no one else is looking at OTX2 in prenatal stress models. I haven't also yet looked at that particular mark. My guess, based on looking at these ELS1 versus ELS2 timeframes, is that we might be in more specific, again, in the midbrain versus hypothalamus and other areas we might look at to this later postnatal period. But it would be really interesting to see if it mediates different stress periods. The other thing that this question makes me think of is when we're looking at a 2-hit stress model, which I've presented here, it's really important to control as much as possible, and I have to do all my breeding in-house because shipping stress is a huge prenatal stressor, and we actually see that we get more depression-like behavior among animals that we ship in pregnant. So for anyone looking to expand into this type of work, I would caution you to breed in-house and not get shipped pregnant dams.
Yeah, what is it about the sensory environment of the animal that's actually triggering the gene changes? Is there anything known about that? You know,
What aspect of the
somatosensory,
stress?
olfactory, food. You know, what's the mechanism that triggers the gene expression changes?
It's a great question. I did a lot of different work looking at different potential stressors in early development before we sort of landed on this, and most of the time the mice didn't care. I think one of the things that seems to be important that emerged from a lot of that sort of negative data is that the animals were separated from the home cage rather than the mother separated from the home cage. So that changes a lot of the sensory environment, especially the olfactory cues that they're receiving. Regina Sullivan's lab has done a lot of work sort of indicating that if the offspring has any olfactory cues from the mother, it actually buffers the stress response. So I think that is probably a huge component of that. But we can't, at this point, tease apart the nutritional changes because most early life stress paradigms, including this, lead to changes in weight gain, which recover by adolescence and even overtake in some cohorts by adulthood. But we don't know if that 4 hours of deprivation where they're not able to nurse is also playing into that.
All right, one final question.
I wondered if you had any plans to look at gene by environment interactions, if there's different genetic backgrounds that might confer resilience.
I don't have any plans at this point.
Okay, yeah, because there is a heritable component to depression.
So the heritable component to depression is much smaller than compared to other psychiatric disorders.
Sure, yeah.
For schizophrenia, it's 60-70%. For depression, it's more like 25%, which is again one of the reasons that we think it's largely environmentally driven and less genetically driven. The more power we throw at these GWAS studies, the more they are identifying some regions, but and some of the regions, there's a 23andMe paper that came out last year, are some of the genes that had led to OTX2's prediction. So it does seem to be playing into what we know about the genetic components of depression that we're able to model here in the mice even, but I'm not specifically looking at those regions, no.
Great. Well, everyone, thank you.
Thank you.
Cate Peña.